Our next speaker is Professor Ronald Hart of Rutgers University.
I'm going to talk today about induced pluripotent stem cells and how it's really time for the academic scientists, at least, to think about transitioning from rodent models to real human cells to study human disease, and how we've leveraged the vast power of the RUCDR program on campus to take advantage of these technologies. So I'm going to very quickly cover what are induced pluripotent stem cells, how we've devised a scheme for routinely making iPS from patients with specific genetic disorders, and then just give you a very, very short summary of some of the Rutgers projects involving iPS that are already underway. iPS are just induced pluripotent stem cells. They're somatic cells that have been reprogrammed with transcription factors to become essentially identical to embryonic stem cells. By most measures, they are indistinguishable. If you look hard enough, you can find very, very minor differences. But for all purposes that are of any use to anyone, they function identically to embryonic stem cells. They're made by ectopic expression of one of several different kinds of mixtures of transcription factors. I've listed two of the original sets here, the ones from the original Yamanaka papers, and the one from the original paper from Jamie Thompson's lab. They're essentially several lists now of potential factors that can make these cells, and they have the property that almost all of them cross-induce the missing factors from one list to another. These can be delivered by one of several methods, most of them including viruses. The original methods were either retro or lentiviruses. We've gone to all Sendai virus protocols, and I'll tell you about that in a few minutes. And you can also do this very inexpensively using plasmids that contain eukaryotic replication machinery called episomes. There also are some protocols for replacing factors with small molecules. As far as I see at this point, they are not routinely used by many programs and have not been widely accepted yet, but I suspect they will be at some point. And remember, finally, that all pluripotent stem cells in the laboratory are pluripotent and they're not totipotent. They are not capable of forming whole embryos because they're unable to form the extra embryonic tissues such as placentas that are necessary to form whole organisms. There are tricks in mouse to take mouse iPS cells and mix them with tetraploid embryonic stem cell, tissue to make whole organisms, but that's really not possible or ethically allowable in humans as well, so it makes sense that that's not a consideration. And what I'm showing here at the bottom is just an example of a teratoma tumor that's formed from human iPS cells injected into mouse, which when you slice it open and do the histology, you can see all three germ layers showing up in that tumor, proving that they're pluripotent. This, of course, all came from work that was started actually in the late 50s at this point. The paper that's cited in the Nobel Prize was from 1961, but the real first paper that John Gurdon published was here in 1958 in Nature, where he did somatic cell nuclear transplants, as it's now called, to create a whole organism, in this case a Xenopus frog. And I always like to show, this is a recent picture of John Gurdon, who's still scientifically active. I love to show this picture because I'm terribly jealous of his hairline. But he continues to work at now the John Gurdon Institute and does really valuable work to this day. Following up years later on these concepts, which are actually not very widely accepted at the time, a whole group of scientists, mostly at MIT and Harvard, followed up on this after the incidence of human embryonic stem cells and the difficulty in creating human embryonic stem cell line, especially during the Bush years. They did some somatic nuclear transplant in human cells, but they reasoned that the reason that worked was because there were transcription factors in the acceptor cytoplasm that then would be reprogramming the transplanted nucleus to form pluripotency. Based on this concept, Yamanaka went ahead and screened batches of transcription factors that were known to be expressed in embryonic stem cells for their ability to reprogram somatic cells, and successfully reported this in mouse cells in 2006, followed very quickly thereafter by reports in 2007 in human cells, both from Yamanaka's lab and from Jamie Thompson's lab. The use of human iPS cells as opposed to embryonic stem cells gives us a huge list of advantages. For example, this summary from Ricardo Dolmesh, formerly of Stanford, shows that in most disease studies, you can compare lower animals and mouse models, which are it's turning out to be more and more different from human biochemical mechanisms the closer you look. Compared with human postmortem tissues and live human studies that can't be done with much throughput, iPS cells really have all of the benefits of cell culture systems, of mouse models, yet they're culture systems that can be done at high throughput, and they are truly human models. Furthermore, you can use them to capture human genetic diversity. Biologists commonly forget that humans are not mice. They're not inbred, uniformly genetic animals. They're outbred. And so the human population is highly genetically diverse, and the geneticists all know this, and we cell biologists and molecular biologists have ignored this blindly for years. We have now a mechanism for making sufficient numbers of iPS cells from sufficient numbers of donors to at least begin to represent that diversity. And so it's an important part of the consideration when switching to an iPS platform. As a disease model, it has huge numbers of advantages. This comparison from Richterly shows that embryonic stem cells can also be triggered to make many different kinds of cell types where you can set up disease models and tissue-specific culture systems. But iPS cells are just as pluripotent and have the same capacity for forming these differentiated culture models of human disease. Plus, they have the history of the subject from which you got the human sample. They have the genetics of that subject. You may have the family history of that subject, all of which can inform your biochemical processes. So you cannot look at an embryonic stem cell and determine whether it's schizophrenic. There's no way to diagnose an embryo to know whether it's schizophrenic. You can take an iPS cell from a schizophrenic patient and make it a pluripotent stem cell line. So we know all about family history, symptoms, penetrance, all kinds of things that you cannot get from an embryonic stem cell study. Based on these philosophies, we've turned to the Rutgers University cell and DNA, well, actually not called that anymore, RUCDR Infinite Biologics, to set up a program to take advantage of this wealth of collection on campus. The RUCDR has over 500,000 human subjects stored in liquid nitrogen, or at least cells from those subjects, not the whole subjects. This isn't Walt Disneyland here. There's more than 4.5 million samples stored, and they distribute more than 1 million samples per year. It's a huge organization. It hosts collections from a number of agencies. Some of them are listed here. Importantly, many of those programs were done for genetics, and therefore many of these cells have already been screened by genome-wide association study for genome-wide SNPs. This gives you the capacity to go into these collections, find the targeted SNP sites that have been already associated with a disease or disorder, hand pick out cells from the donors that participated in the GWAS to have specific SNP patterns and make IPS and do the biochemistry and study those functions. Based on this, we were awarded the funding from NIMH to set up the stem cell research that supports NIMH operations and NIMH-funded researchers. So now NIMH-funded researchers who do IPS creation are encouraged by their granting agency to send their cells to RUCDR for archiving and further distribution. They're also encouraged to send any source cells that may be useful in making IPS cells down the road. And there has been programs in collaboration with some of the NIMH-funded researchers to differentiate and produce some of the product cells as well. On campus and many other funded programs, we've also used this same strategy to select high-risk SNPs and to create genetics models for other programs that I'll mention a few of these in detail. Finally, we also store and distribute the IPS collection from the NIH Center for Regenerative Medicine, which includes many disease models and a large group of controls as well. We've already produced more than 150 IPS lines. I put greater than at this point because I've lost track of how many lines the staff has created, and I'm not directly part of that program. This is from more than 60 human subjects from a wide variety of programs, including some of them listed here, schizophrenia with outside NIMH collaborators and others, autism with some collaborators and others. We've done addiction genes, alcoholism genes, Tourette's, and in my most extreme case that we did in my lab, we took a blood sample that was frozen over 20 years ago from an Alzheimer's patient that died at age 86 20 years ago, thawed it, and was able to make a IPS line that turned out to be ApoE E4E4. So even all these blood samples that are in long-term storage are still useful to us. And it shows the power of this repository. For all of these IPS lines, we go through a very rigorous set of characterization protocols. In this case, colonies that are derived from fibroblasts are staining positive for TRA160 and OCT4. They're also double positive by FACT, showing that every cell is positive for both markers. Gene expression studies show that their expression pattern is indistinguishable from embryonic stem cells. We can do embryoid body staining to show that they generate all 3 germ layers. And we can do this from either fibroblast-derived sources or lymphocyte-derived sources, as I'll describe in a moment. And we get the same kinds of products. This is an alkaline phosphatase-based assay as well. And finally, we can produce functioning differentiated cells. This example is neurons, as you'll see more of in a moment. They're 2UJ1 positive and MAP2 positive, showing a mature neuronal phenotype, and they can be stimulated to have electrical activity as well. Our main pipeline is shown here. Since most of our cells are blood-derived cells, we found that the most efficient process takes advantage of a cell type called cryopreserved lymphocytes. They're essentially the white blood cells that were taken off the buffy coat from the tubes when these first came in as raw bloods into the RUCDR. The traditional pipeline in RUCDR is to take those CPLs, infect them with EBV to make lymphocyte cell lines, or LCLs. What was commonly done, luckily, was to save the leftover CPLs as a backup in case the EBV infection didn't work. And that's what we've been using primarily at this point. There's no EBV in them, which makes it just one step cleaner for our purposes. We thaw the CPLs. We go through an enrichment process where we magnetic bead remove other cells besides the CD4 positive T cells. We enrich those T cells by activating them with 2 cell surface markers and IL-2. They expand and they activate. We then infect them with commercial Sendai virus preparations. This is the Cytotune kit from Life Technologies that's licensed from a Japanese company that manufactures them. And then they produce the pluripotent colonies within about 2 to 3 weeks very efficiently. And this entire process has been published in an ebook that's available online if you wanted to see every detail of our process. Again, these lymphocyte-derived cultures are able to make all 3 germ layers in embryoid body formation. We've been trying to get away from the teratoma assay, and many stem cell researchers have, because it's slow and expensive. And really, it's not very helpful. You don't get a good you don't get a good failure rate off of the teratoma. You never know why you get a failure is the problem. So we've gone to an embryoid body assay, which is a little simpler and faster and cheaper. But then we also go through another series of assays as well. We do karyotyping, and of course that only looks for very gross genomic abnormalities. And every line is karyotyped and compared back to its original parental line. We also have a 96 SNP RUID system that is used for identity confirmation. So with this 96 SNP pattern, and they're just color-coded here to show you how identical they are, all of the IPS lines turn out to be identical to the parental line in the top row. This is being run on every single sample that's in the RUCDR, and it confirms the identity of every single sample we have genetically. Furthermore, we do microanalysis, and this is a useful algorithm that reduces a microarray's worth of data down to a very small number of measurements, in this case 2. So we reduce the 30,000 or more genes that we've assayed down to 2 dimensions, pluripotency and novelty, using a unique algorithm developed by a group at UCSD. This is called the Pleura Test. And what it does is it partitions the gene expression pattern into one of 4 quadrants, and only 2 of them are really informative here. The red cloud in the background represents the gene expression patterns from 223 embryonic stem cells from a wide variety of laboratories around the country and 41 IPS lines. And the blue clouds over here represent over 450 differentiated lines as comparisons. Our lines are shown as the dots. The green dots are all embryonic stem cells or IPSs that we've manufactured, versus the source cell lines in yellow over here, either T cells, CPLs, or fibroblasts, or differentiated products of the iPSC, here shown in orange. These are neural stem cells. So we can very quickly classify the IPS following a broad pattern of gene expression. Furthermore, using the Sendai virus vector is crucial because we do not integrate virus into the genome of the IPS line, like a lentivirus or a retrovirus would. The genomic DNA of the iPSC lacks a detectable Sendai sequence assayed by PCR. We could have detected if it was there. We spike in 1/10 of a copy per genome, and we can clearly see that. We look at the RNA viral load, and before infection, there is some background level. This is the log scales. This is basically undetectable. We see a million-fold increase upon infection that decreases by about 11 or 15 passages to essentially background levels, no virus detectable whatsoever. We can make differentiated cells from these cultures, and this is now outside of the RUCDR group. In one of my projects, we were able to make dopaminergic neurons. The red staining is tyrosine hydroxylase, which is specific for dopaminergic synthesis. And you can see that they co-stain for both TOJ1 and MAP2, indicators of neurons, and synapsin, which is indication of the synapses formed in these cultures. They also show some contaminating neuronal types. There's 5-HT for serotonin and GAD6 for glutamatergic. And these are post-mitotic cells. They do not stain for nucleic acid replication. They are functional. We see very nice spontaneous action potentials, elicited action potentials, and postsynaptic currents, showing that there are synapses in these cultures. When we culture them on rodent glia cells, we get much more robust results for this, of course. We also can see calcium spiking spontaneously, and I'm not showing it here upon induction as well. As a couple of examples of projects on campus, in one example studying schizophrenia and one gene associated with a subset of schizophrenic patients, we see that the number of processes coming out from these differentiated neurons is reduced when that gene is overexpressed, as it is in schizophrenic patients. And if you count those, you see a reduced branching in those cells, so we can replicate this lack of connectivity in a schizophrenic neuron. In another example, we can do neurotoxicology. This is work by Jason Richardson at the EOSHI on campus. He's looking at chloropyrifos, a common insecticide, and shows that at the neural stem cell stage, we do not see toxicity, but we do see expression of some of the target enzymes in these cultures, and we see changes in gene expression that I'm not going to discuss in detail. Finally, we've done some three-dimensional surface construction. This is in collaboration with the biomaterials groups on campus and the bioengineering groups on campus. We can create a three-dimensional surface by various chemistry with various dimensions. And we find, for example, that neural cells induced from iPS cells adhere more selectively to these three-dimensional engineered surfaces than to a two-dimensional culture surface. And we see more functional properties in these cultures than in a two-dimensional culture. So very briefly, just to wrap up, we've turned to the capacity to use human cells to study human disease, particularly in neural disorders and behavioral disorders. We obtained functional neurons from these subjects. You can't get neuronal donations from live human subjects. We've asked, and they won't agree. We can test mechanisms of human diseases in human-based models. And we can do toxicology in the appropriate cell types with known genetic backgrounds, if that's appropriate. And we can develop engineered materials for performing some of these cultures that may improve our outcomes. This is a product of both labs at Rutgers, and this is just listing my lab and the RUCDR group. You'll see Jen Moore listed twice because she's a part of both groups, and she's a key person in this process. And our collaborators are listed here, mostly either Rutgers or UMD&J based collaborators. And after we're done, I'll be happy to take any questions. Thank you.